Hello everyone, my name is PUSPITA Saha. Currently, I am working under the supervision of Dr. Moha Maulik and Dr. Maulinath Acharya at BRIC in IBMG. Today, I'll be presenting the highlights of my work on understanding how the dysfunction of the major astroglial gap junction protein connexin 43 leads to the impaired gap junction communication with relevance to Alzheimer's disease. In recent years, astroglial dysfunction has emerged as a common pathophenotype alongside the neurocentric research approaches in Alzheimer's disease. In an AD brain, the astrocytes are characterized by the loss of the gap junction activity. In various studies conducted in postmortem AD brains and in transgenic AD mouse models, it has been shown that there is an accumulation of the reactive astrocytes in vicinity of the amyloid beta plaques, and in rodent cultures, it has been shown that amyloid beta impairs the functions of these astrocytes of these gap junction proteins. But the mechanistic basis of this process has remained unclear, and along that line, we proposed our study, and we conducted our study in an in vitro cell culture model of primary human astrocytes characterized by the expression of connexin 43. And we treated these cultures exogenously with the synthetic fragments of amyloid beta peptides. And we found that upon increasing exposure to amyloid beta, there is a decrease in the gap junction communication. When we asked that how this disruption in the gap junction activity is happening, we found that amyloid beta increases the intracellular accumulation of CX43, while in the untreated cultures, the CX43 protein is still present on the surface, lining the cell-cell borders. When we asked where this intracellular pool of CX43 is headed, we found that amyloid beta induces the ER sequestration of the connexin 43. This prompted us to question that there might be an accumulation of misfolded CX43 which gets entrapped in the ER and fails to traffic towards the surface. And we found in our proteasomal inhibitor treated cells that amyloid beta induces the ER stress associated degradation of CX43. Overall, we show here that amyloid beta leads to the impairment of ER homeostasis. So we overexpress the ER resident protein ERP29, that is the canonical chaperone of CX43, and we found that ERP29 could rescue CX43 from the amyloid beta induced ER arrest. To reinforce the credibility of our findings, we performed a similar study with 4-PBA, that is a chemical chaperone and an FDA approved drug. And we found that 4-PBA could also reverse the intracellular retention of CX43, leading to the consequent restoration of the gap junction activity. Overall, we show here that amyloid beta-induced ER stress and reduced CX43 ERP29 interaction leads to the impaired gap junction activity, and highlight the importance of ERP29 and 4-PBA in the treatment of Alzheimer's disease neurodegeneration, both of which are able to restore the gap junction activity. Thank you.